Bismillahirrahmanirrahim. Assalamu alaikum. I am Muhammad Hassan Ashraf, lecturer zoology from Faisalabad campus. We are going to discuss our lecture number 21, the biotransformation, and this is the continuation of the previous lecture of the biotransformation. Here we will start with the biotransformation sites. The mainly, as we discussed in the previous lecture, the two phases of the biotransformation, the phase 1 reaction and phase 2 reactions. These phase reactions takes place in different body organs, and the main sites for the biotransformation are liver, kidneys and lungs, and skin, intestine, testes and placenta. From all these organs, livers are the most important and the major sites of the biotransformation. So liver is primary biotransforming organ due to its large size and high concentration of biotransforming enzymes. The kidneys and lungs, they perform biotransformation about 10 to 30% of the liver's capacity. Means liver have maximum capacity. After liver, the two organs, kidneys and lungs, constitutes in the mechanism of biotransformation. Then the skin, intestine, testes and placenta, they have a low capacity of biotransformation. Primary biotransformation site, the liver, because mainly biotransformation takes place at liver, so it is also called as the primary biotransformation site. Since the liver is the primary site for biotransformation, it is also potentially vulnerable to the toxic action of a xenobiotic that is activated to a more toxic substance. Within the liver cell, the primary subcellular components containing the transforming enzymes are the microsomes, which is the meaning small vesicles. These microsomes are the small vesicles of the endoplasmic reticulum and the soluble fraction of the cytoplasm, the cytosol. The mitochondria, nuclei, and lysosomes contain a small level of transforming activity. The microsomes, these microsomal enzymes are associated with most phase 1 reactions. Glucuronidation enzymes are also contained in microsomes, and because glucuronidation is a phase 2 reaction, so mostly phase 1 reaction enzymes are present in microsomes along with some of the chemicals for the phase 2 reactions of biotransformation. Cytosolic enzymes are non-membrane bound and occur free within the cytoplasm. They are generally associated with phase 2 reactions, although some oxidation and reduction enzymes are contained in the cytosol. The most important enzyme system involved in phase 1 reaction are the cytochromes P450, also called the cytochrome P450 system or the mixed function oxidase system, MFO, mixed function oxidase. It is found in microsomes and is responsible for oxidation reactions of a wide array of chemicals. Susceptibility of the liver. The liver is particularly susceptible to damage by ingested toxicants because it biotransforms most xenobiotics and receives blood directly from the gastrointestinal tract. Blood leaving the gastrointestinal tract does not flow directly into the general circulatory system. Instead, it flows into the liver first via the portal vein. This process is known as the first pass. Blood leaving the liver is eventually distributed to all other areas of the body. However, much of the absorbed xenobiotics has undergone detoxification or bioactivation. The liver may have removed most of the potentially toxic chemicals, On the other hand, some toxic metabolites are highly concentrated in the liver. So liver is the site which is most susceptible to the influence of toxicants, particularly those toxicants which enter in the body through the oral route. The food along with the toxicants or some drugs ingested and absorbed through the small intestine firstly enter into the liver for the detoxification. The liver is the site which is maximum influence of toxicants. This faces the maximum force of the toxicants, and then the liver also changes or biotransforms those toxicants into the products which can be eliminated from the body. Modifies fires of biotransformation. The relative effectiveness of biotransformation depends on several factors that can inhibit or induce enzymes and dose levels. These factors include the species in which the biotransformation is taking place, age of the organism, the gender, either it is male or female, genetic variability. Some have more stronger genetic makeup which prevents their body from the effects of the toxicant. Some have weak genetic makeup which allow the effect of the toxicants. Nutrition, disease, and exposure to other chemicals. All these factors they collectively modifies the mechanism of biotransformation. Firstly, the species. Differences are termed selective toxicity. There are the different species behaves differently against same toxicant, and this effect is called as the selective toxicity, because it is not necessary that if a chemical is toxic for one species, it is also toxic for other species. It is not always a factor. Differences are termed selective toxicity, which refers to differences in toxicity between species similarly exposed. For example, the pesticide malathion in mammal is biotransformed by hydrolysis to relatively safe metabolites, but in insects it is oxidized to meloxicam, which is lethal to insects. So different species behaves differently and biotransforms same chemical differently in different body organs. The second factor is the age. The age may affect the efficiency of biotransformation. In general, human fetuses and newborn have limited abilities to carry out xenobiotic biotransformations. This limitation is due to inherent deficiencies in many of the enzymes responsible for catalyzing phase 1 and phase 2 biotransformations. While the capacity for biotransformation fluctuates with age in adolescents, but by early adulthood, the enzyme activities have essentially stabilized. The aged also have decreased biotransformation capability. That's why they are more susceptible to diseases and the effects of the toxicants. Next is the gender. The gender may influence the efficiency of biotransformation for specific xenobiotics. This is usually limited to hormone related differences in the oxidizing cytochrome P450 enzymes, because male and female both have different hormones. They have at least some secondary hormones which are different in both male and female, and due to that difference they can behave differently in the mechanism of biotransformation of a particular toxicant. Genetic variability. Genetic variability in biotransforming capability accounts for most of the large variation among humans. In particular, human genetic differences influence the phase 2 acetylation reaction. Some persons have rapid acetylation, the rapid acetylators, while others have a slow ability to carry out this reaction. The most serious drug related toxicity occurs in those who have slow acetylators, often referred to as slow metabolizers. with slow acetylation is so slow that blood or tissue levels of certain drugs or phase 1 metabolites exceed their toxic threshold. Here are some examples of drugs that build to toxic level in slow metabolizers with specific genetic related defect in biotransforming enzymes. The name of the drug is isoniazid, drug category anti-tuberculosis drug. Its metabolic defect is slow acetylation, and the toxic effects observed are the nerve and liver damage. Next is the drug is hydralazine. It is anti-hypertensive drug, and its metabolic defects includes defects in monooxygenase enzyme, and due to this metabolic defect the toxic effects of these drugs are excessive fall in blood pressure. Then dapsone, it is antibacterial agent. Its metabolic defect is slow acetylation, and this slow acetylation leads to systemic lupus erythematosus. Then primaquine is anti-malarial agent, and its metabolic defect is defective G6PD, and the toxic effect are acute hemolytic anemia. Then the next factor is which modifies the process of biotransformation is the nutrition. Poor nutrition can have a detrimental effect on biotransforming ability. Poor nutrition relates to inadequate levels of proteins, vitamins, and essential minerals. These deficiencies can decrease a person's ability to synthesize biotransforming enzymes. Many diseases can impair an individual's capacity to biotransform xenobiotics. For example, hepatitis is well known to reduce hepatic biotransformation to less than half of its normal capacity. Prior or simultaneous exposure. Prior or simultaneous exposure to xenobiotics can cause enzyme inhibition and enzyme induction. In some situations, exposure to a substance will inhibit the biotransformation capacity for another chemical due to inhibition of specific enzymes. A major mechanism for the inhibition is competition between the two substances for the available oxidizing or conjugating enzymes. The presence of one substance uses up the enzyme needed to metabolize the second substance. So when there is a simultaneous exposure, the biotransformation can be modified by this simultaneously exposure to different type of toxicants. Exposure to other environmental chemicals and drugs. Enzyme induction is a situation where prior exposure to certain environmental chemicals and drugs results in an enhanced capability for biotransforming a xenobiotic. The prior exposures stimulate the body to increase the production of some enzymes. This increased level of enzyme activity results in increased biotransformation of a chemical subsequently absorbed. So, if the chemical exposure increases enzyme activity, that will definitely increases the effect or biotransformation of the toxicant. Here are some examples of enzyme inducers. They include alcohol, isoniazid, polycyclic halogenated aromatic hydrocarbons, for example dioxin, phenobarbital, cigarette smoke. The most commonly induced enzyme reactions involve the cytochrome P450 enzyme. So these are the different inducers which enhances the activity of enzymes. They increases the activity of enzyme, and some of them acts as toxicant as well. The dose level. Dose level can affect the nature of the biotransformation. In certain situations, the biotransformation may be quite different at high doses compared to low dose levels. This difference in biotransformation contributes to a dose threshold for toxicity. The existence of different biotransformation pathways can usually explain what causes these dose related differences in biotransformation. So dose also does matter. If the low dose of a chemical is injected or administered, then the biotransformation can easily takes place. If the dose is much higher than the threshold limit, then the biotransformation process is not easy. It will take more time and not all the toxicant is biotransformed. At low doses, a xenobiotic may follow a biotransformation pathway that detoxifies the substance. However, if the amount of xenobiotic exceeds the specific enzyme capacity, the biotransformation pathway is saturated. In that case, it is possible that the level of parent toxin builds up. In other cases, the xenobiotic may enter a different biotransformation pathway that may end up producing a toxic metabolite. So the dose does matter. The higher doses will saturate the enzymes and inhibits their ability to biotransform that toxicant. If the toxicant is not biotransformed, then it can directly cause its harmful impacts on different body organs. So dose level should be within the threshold limit. If it is exceeds from the threshold limits, then it is difficult to detoxify or biotransform that drug. Here are some examples. An example of dose-related differences in biotransformation occurs within acetaminophen, the Tylenol. At normal dose, here is a comparison between normal dose and higher doses. At normal dose, that is about 96% of acetaminophen is biotransformed to non-toxic metabolites by sulfate and glucuronate conjugation. By this, at normal doses, this is biotransformed by the phase 2 reactions. About 4% of the acetaminophen oxidized to a toxic metabolite. That toxic metabolite is conjugated with glutathione and excreted. So if this dose is at its normal level, then 96% plus 4%, 100% will be biotransformed or excreted out from the body. But if a dose administered which is about 7 to 10 times the recommended therapeutic level, then the sulfate and glucuronide conjugation pathways become saturated and more of the toxic metabolite is formed. The glutathione in the liver may also be depleted so that the toxic metabolite is not detoxified and eliminated. It can react with liver proteins and cause fatal liver damage. So the normal dose, if there is, then the normal metabolic mechanism removes the toxic effects. If that dose exceed from the normal limits, then the normal metabolic functioning cannot be performed. It cannot be transformed or can be excreted from the body. As a result, that drug start to damaging the different body parts. These are the references. So this was all about the biotransformation mechanisms. Thank you. Allah Hafiz.